This is the TPD app homepage. From here, you can view, download, or delete your analyzed data. See the status of analyses in progress or launch new analysis. Click on Plate Maps to view your plate map library. In the plate map library, you can download or edit an existing plate map or create a new plate map. The green icon indicates that your plate map is complete and ready to be used for analysis. Incomplete plates will have a red icon. In the plate map editor, the fields at the top are modifiable and apply to the whole plate. To save the plate map, including any changes, select Save. Save As will save as a new plate map. There are 3 well type options. Sample, which includes cell line expressing high bit edited target, and large bit experimental compound and detection reagent. Background, which contains the parent cell line expressing large bit, but no high bit edited target and detection reagent. And blank wells, which are not used in analysis. The target field applies to sample wells only. The cell type field applies to both sample and background wells. Make sure the sample cell type matches the background well cell type to ensure correct background subtraction from sample wells. The sample wells have a compound input box with a concentration unit dropdown. The same concentration unit must be applied to all sample wells that are part of a concentration series. A concentration series can be applied to multiple wells by selecting concentration series, inputting the highest concentration, the dilution factor, then dragging the edges of the first well either vertically or horizontally across all wells in the series. A minimum of 3 compound concentrations are needed for analysis. Additionally, a no compound or DMSO control well is required for analysis. This should be input as a sample well with 0 concentration. Make sure to save your plate when you are done. To start new analysis, select New Analysis from the app homepage. You will see an overview of the steps in the workflow. First, choose your source data file. This is the raw kinetic data you selected from a connected GLOMAX instrument. Next, name your analysis. Next, you can either create a new plate map or select a previously created plate map. The next step is normalization. You can either skip normalization, normalize to a pre-read plate, or normalize to an iteration from the kinetic data file you chose in the source data step. If you want to normalize a pre-read plate, select the pre-read plate data file from a connected GLOMAX. To normalize to an iteration from your kinetic data file, select Iteration and input the iteration number. Note that analysis requires a minimum of 50 iterations. In the data check step, you can review fractional RLU by target/compound combinations before launching your full analysis. In this step, you can download the raw kinetic file, the normalized kinetic file, and the pre-read plate file if used. Monitor the status of your analysis from the TPD app homepage. When your analysis is complete, you can view graphs for all target compound combinations. The Dmax includes Dmax 50 confidence interval, Dmax 50, and model fits. Hover over data points to see more specific information. Under the Metrics tab, you can see graphs of the degradation rate, duration at Dmax and Rmax. If the target protein does not recover, the recovery graph will be blank. All data may be downloaded as a zip folder from the app homepage. Files in this downloaded zip folder include the normalized kinetic data, the plate map, a pre-read plate if used, and the raw kinetic data. Additional results include AUC 50, Dmax 50, and metrics. More information interpreting these data can be found in the TPD datasheet.